Good morning, everyone, and thank you for coming to this seminar. I'm Brenda from the Scientific Support Specialist team that is responsible for Asia Pacific region. There's three of us in this team, and yeah, I'm one of them. In today's talk, it will be separated into four different sections. We will first start off with a brief introduction to IHC before proceeding to antibody selection. I will then move on to some tips on transitioning from manual to automated IHC before ending this talk with some troubleshooting tips. So without much further ado, let's start with the first section. So what is IHC and why is IHC commonly used? IHC stands for immunohistochemistry, and as its names imply, a primary antibody is used to detect the antigen or target protein in the tissue. The signal will then be visualized by either a reporter molecule conjugated directly to the primary antibody or by binding to a secondary antibody with a reporter to amplify the signal. So because of this, IHC is an important tool for researchers to study protein expression and localization, and it is widely used in basic and clinical research, and also more recently in spatial biology, which is a growing field. So what exactly is spatial biology? Spatial biology essentially looks at the different expression of multiple markers or proteins within the context of a tissue to really get a feel of where they are and how they are behaving relative to each other. It is an imaging-based analysis detecting multiple markers on the same cell. Therefore, you can really get the real-time physiological insights into what's happening and the relationship of this protein within a tissue. It is also commonly used in cancer oncology, immuno-oncology, and neuroscience research. In cancer research, this allows for deep dissection of the tumor microenvironment, precisely mapping the infiltration patterns of a range of immune cell subsets and their proximity to cancer cells. It is also frequently used in discovery with the hopes of bringing precision medicine to clinical use. So at the discovery stage, clinical researcher would do spatial phenotyping of a large number of biomarkers to determine if there's any relevant biomarkers or signatures. So for example, do we see a glimpse of interaction maybe before or after the treatment that could give some type of indication of what is happening? And once a certain markers are identified, it will then proceed on to the translational phase. So this is where you will now have an idea of a pathway or signature that you do see interacting or expressing differently based on the discovery stage. A smaller number and more targeted panel would be used, but sample throughput would be increased to drill down to a suitable biomarker. And at this stage, the goal is to see if this can help to guide clinical trials to figure out which therapy a patient may respond to. And eventually, what we really hope to achieve is that we can take this to the clinic where precision medicines can be applied. And therefore, as we progress from the discovery stage to the clinical, you can see that the number of plex decreases while the throughput, which is the number of samples that you are running, actually increases so that it can be applied to more patients. To look at multiple markers, multiplex IHC is commonly used in spatial biology. So this is because it allows for simultaneous detection of numerous biomarkers within a single tissue section, preserving the spatial contact architecture and specifically the intracellular interaction. So for example, in this image is 7-plex staining. So you can see that the immune cell, which is the macrophages, are denoted by this yellow color, the CD68. and CD3 is for your T cells markers, and pan-CK is actually for the epithelial cells. So you can really see how the different cell types are in relation to each other. And you can also compare this to the expression of PDL1 or PD1, which is an indicator of the cell proliferation. So this really gives you a context of the spatial relationship of different proteins within the cells. Multiplex IHC can be simple or complex depending on the number of markers you intend to look at. So if you are doing a lower plex, for example, just 3 to 4 markers, the standard IHC would usually be sufficient. So for example, if you are using a chromogenic detection, whereby an enzyme is used and you add a substrate for color detection, this can usually be done for up to 3 colors. Or alternatively, you can also use a fluorophore conjugated primary antibody, or in this image is showing indirect staining. So you are using a secondary antibody to then detect the fluorescence. So this is more of the standard IHC when you are doing lower plex. But if you are looking at a higher plex with multiple markers, so for example, if you are looking at 8 or 10 targets, it may be hard to use the standard IHC protocol due to spectral overlap of the fluorophores. So to overcome this, there are several ways that higher plex IHC can be done. The first is to use fluorescently labeled primary or secondary antibodies. So this is just a normal IHC method, but you can couple it with technology such as TSA. So this is commonly used for cyclic immunofluorescence or cell dive which is a platform by Leica. So what they do is that they employ iterative cycles of staining with antibodies, imaging, and then stripping or quenching the signal before staining for the next target. Alternatively, you can also use antibodies that are labeled with oligonucleotides or nucleic acid barcode instead of fluorophores. So this would include technologies such as Codex, which use a cyclic staining, or you can also use JORC DSP by NanoString. And last but not least, the antibodies can also be labeled with heavy metal isotopes, allowing simultaneous detection through imaging mass cytometry platforms. So in this next section, I would like to briefly cover the benefits of using ABCAM antibodies. You might ask why choose ABCAM as the providers for IHC antibodies for your experiments. So the first reason is that based on site antibody data analyzed in September last year, ABCAM has the #1 number of cited antibodies that were used in IHC experiments. And also all our antibodies actually undergoes rigorous validation before they are made available. Our largest and faster growing collection of recombinant antibody is our rabbit monoclonal antibodies, which is raised using our patented RedMap technology. We currently have more than 34,000 RedMap available, of which over 4,000 are IHC validated antibodies available in carrier-free format, which allow for easy adaptation for downstream application or conjugation process. So the reason for choosing rabbit is really due to the intrinsic rabbit's ability to mount an immune response against a broader range of compound, and thus providing a higher affinity and specificity antibody than that using mouse or rat. We also further apply our proprietary recombinant technology to our rabbit monoclonal to produce recombinant RedMap antibodies with improved consistency, reproducibility, sensitivity, specificity, and scalability. And last but not least, as I mentioned previously, every RedMap antibody is actually screened and tested in multiple applications and species to ensure the highest quality possible. And all this information are usually available on the data sheets, so you can refer to it before starting your experiment or before purchasing the antibody. So for our IHC standard validation, we use an in-house tissue microarray comprising of 16 normal human tissue as well as the cancer tissue. And then we also include a normal mouse and rat tissue. So in this tissue microarray, it includes both the positive and negative control to ensure that the staining is specific. We'll also cross-reference it to the literature to see if the staining patterns align. And if it does not align, then the antibody would not be recommended for IHC application. Another key highlight on our quality control is that we have been using knockout cell line models to validate our antibodies so that you do not have to do so. We currently have around over 4,500 KO validated clones available on our website. So whenever possible, we will do a Western blot with the KO cell line. So for example, in this image, you can see that for the DR5 KO cell line, the cleaved caspase 7 bands is missing, which indicates that this antibody is specific to caspase 7 caspase 7 protein. Or in other instances, we also use knockout cells in IHC application validation. So these are actually cell block, and you can see that PDL1 is expressed and stained in the parent cell, but in the knockout cell line, the staining is no longer observed, which helps to further show the specificity of this antibody. And we understand the importance of selecting an antibody that is specific and sensitive to your target. So apart from our routine testing, we also recently introduced a biophysical characterization step in our QC process. So this will be using techniques like mass spectrometry as well as HPLC to ensure that the antibody is what it is and that there are no aggregation or contamination before it is made available. And we also introduced our enhanced validation program to go above and beyond our already rigorous standard validation. So for this enhanced validation, the antibodies are profiled through extensively specific and sensitive testing on up to 600 individual relevant disease cases. So I will just share more on regarding the enhanced validation in the next few slides. So one example is our anti-Claudin-6 antibody. So we also included the cancer-specific tissue microarrays, such as for lung cancer, ovarian cancer, and testicular cancer, in addition to the standard tissue array that we use in our routine QC. And after staining for them, we also analyze the expression of Claudin-6 expression in this cancer tissues, and then we came up with the expression level of the Claudin-6 in the different tissues so that you can refer to it, let's say, when you are choosing the antibodies and to decide which tissue cancer cells or to use as your positive control. And in another example for EGFR variant 3, which is a constitutively active mutant form of EGFR observed in 30% of glioblastoma cases, you can see that in this first tissue sample, there is lesser EGFR3 variant expressed, but the wild-type EGFR is expressed quite in abundance, and whereas in this second tissue, glioblastoma cancer tissue samples, you can see that there's more EGFR3 variant that is being expressed. And we also included placenta tissue, which was used as a negative control. So you can see that there's no EGFR3 variant present, whereas EGFR can be seen. So this really helps to show that this antibody is specific for the variant 3 of epidermal growth factor receptor, and that there's actually no cross-reactivity with the wild-type EGFR. And we also further perform a sequential staining of human malignant glioblastoma tissue to further confirm the specificity of this antibody. So in this figure A is first stained for the EGFR3 variant, but without any wild type EGFR. B is for EGFR wild type only. C is the negative control, so it's just IHC diluent without any antibodies. And finally, D is where the tissue samples are stained for both the variant and EGFR. And so this further helped to confirm the EGFR 3 variant antibody specificity in the duplex co-localization assay. So after we did all this testing, they are actually compiled into a comprehensive validation data pack that encompasses the relevant research and clinical settings for the targets in question. And currently is available for these targets, and we are currently preparing the data for PDL1 and CD73. So yeah, you can take a look at this on our website as well. And beside our extensive IHC validation, we also have the largest catalog of recombinant IHC validated antibodies, including the conjugated and carrier-free format, to provide additional flexibility for your experiments. So we have currently over 8,000 conjugated antibodies, of which 800 plus are IHC validated for flexible assay design. But let's say if the fluorophore that you are interested in is not available for the antibody of interest, you can also consider doing the conjugation yourself. So for this, we have our carrier-free antibody that does not contain any BSA, sodium azide, or glycerol. And importantly, these carrier-free antibodies are supplied at an optimal concentration of 1 milligram per ml so that it can be used directly with our lightning link conjugation kits or the conjugation kits from other suppliers. So we currently have over 50 labels, including fluorochromes, biotin, enzyme, such as HRP, oligonucleotides, and metals available for our lightning link conjugation kits. So you can use these carrier-free antibodies directly without any further purification or concentration. So this really allows for a greater flexibility in assay design, especially if you have a specific antibody clone that you are interested in. So apart from these carrier-free antibodies, we also understand that for multiplex staining using the traditional methods, it's important to have antibodies from different species. So we have our chimera antibodies, we have the same specificity of the RAMAT antibodies, but they are grafted onto a different species backbone. So in this example, you can see that so the green color is the variable region where the antigen is binding, and then we graft this, we change the FC receptor region into a different species so that now the antibody is actually raised in a different species backbone. So this allow you to keep the same binding characteristic and kinetics of the recombinant rabbit monoclonal antibody, but using a different species backbone to allow for greater flexibility. So as since you can now use the same antibody in different model system. One important thing to note when using chimeric antibody is that we usually advise to use pre-adsorbed Fc reactive secondary antibodies to prevent any cross or unspecific binding. And last but not least, The reason for choosing our antibodies is that we have a strong relationship with spatial biology platform partners and experts that allow for external validation of our clones on this platform. So we have partnered with NanoString on their GeoMix digital spatial profiler platform, which used to be a 570 plex, but it has recently turned into a 1200 plex. So NanoString has actually shared with us a list of the antibodies where they have tested on and that work for them and build it into a panel. And we also have another collaboration with Prof. Ite Tirosh from Weizmann Institute, which is a very prominent figure in spatial biology, especially in oncology and neuroscience related fields. So he published a paper in Cells where he says our antibody works well with Akoya Phenocycler Codex platform, and he also shared this with us. We also have another collaboration with Professor Greg Hannon from Cancer Research UK, Cambridge Institute. So he uses the Bio2 Hyperion, which is a metal-based imaging mass spectrometry system. And so he screened close to 300 antibodies from us as part of the study, and he has also shared this information with us. And last but not least, we also cooperate with Leica on their self-dive platform. And so far, they have validated over 100 immuno-oncology and neuroscience-related antibodies on their platform. So what we did is that we take all this information and then we compile these into this antibody list that is available on our website. So this can help to save time and cost in early stages of building a panel, since now you know which antibodies will work well with which platform. And this information is especially helpful as some of these technology are relatively new and there are not much publication available yet. So you can use the filters that are located at the top to select for the imaging platform that you are interested in, as well as the target that you are interested in. So we have also included the recombinant antigen retrieval condition and method that can be used so that you can use this as a starting point and you do not have to go around testing for which retrieval method that is better for this specific antibody. And of course, if the antibody the target of interest that you are interested in does not have an antibody available in this list, you can also just do a general search using the search bar by keying in the target of interest. And then you can use the filters located on the left to filter the antibodies based on their application, reactive species, and conjugation, for example. So I'd like to point out that you might want to look for these different banners, such as the KO validated banner, so that you know that this antibody has been tested in KO knockout cell line. And let's say if you are using Leica's Bond machine or automated stainer, then you can keep an eye out to see if the antibody has this Bond RX validated banner. So we normally test our antibodies using manual staining by incubating slides at 4 degrees overnight. But however, if this Bond RX validated tag is present, it means that we also have tried it using automated stainer and by incubating the antibody at a higher temperature. I would also like to quickly share that we classify our IHC application based on sample types instead of the detection method. The two most common sample types would be paraffin embedded tissue sections, so IHCP, or frozen tissue section, which is IHCFR. There are also some other tissue type samples, such as your free-floating frozen tissue sections or the whole mount tissue sections. And the final application would be MIHC, which stands for multiplex IHC, where two or more antibodies are used simultaneously. So if you are looking at doing a multiplex IHC, then please keep a lookout for the MIHC tested application. So you will know that this antibody has been tested in a multiplex settings. So we currently have around 390 plus recombinant antibodies that are tested to work with Leica Biosystem BondMax using the OPA reagent from Akoya Bioscience in lower plex staining. So for all this information, we usually will include the antibody concentration used as well as the incubation timing and the antigen retrieval method available on the product data sheet. And last but not least, just to quickly summarize the different platforms and the type of antibodies that are suitable. So if you're using a oligo conjugation base such as the Akoya or NanoString, you can choose our IHC carrier-free antibody and conjugate it to the oligonucleotide, either using our lightning link conjugation kits or the conjugation kits from the platform providers. And for immunofluorescence techniques such as the Akoya or cell dye from Leica or Lunapause from Comet from Lunapause, you can consider using our standard formulation IHC validated antibody and couple it to TSA or OPA reagent for detection. For Leica cell dye and Lunapause Comet, you can use conjugated antibodies, which would be suitable for the downstream detection. And last but not least, for imaging mass cytometry, you can also consider choosing our IHC carrier-free antibodies and conjugating them to the metals. So I'd like to quickly share that we do have a multiplex IHC detection kit using TSA amplification. So what this kit does is that it helps to conjugate a HRP to biotinylated antibody using a click reaction. Then after that, you can use this antibody to stain your tissue samples and add on the TSA reagent so that the fluorescently labeled tyramide can be covalently bound to the tyrosine residue on or around the protein of interest. So the TSA reagent is not provided in this kit. You will need to get it separately. But once this TSA the tyramide has been deposited, it will then be quenched by adding this HRP blocking buffer to quench any remaining HRP activity, and then you can repeat this process again with another antibody conjugated to HRP. So what this kit is good for is that since you do not use antigen retrieval step to eliminate the antibodies from the previous round, it's actually suitable for both frozen and paraffin tissue sections. And yeah, for this part, this image actually shows a nice overview of the different recombinant antibodies that you can use for the different platforms and methods. And the number of antibodies shown here are the total number of antibodies available for each category, instead of just IHC validated antibodies that I shared previously. So in the event that your target of interest does not have an antibody for which the application or species is validated for, you can also reach out to us as it might be eligible for our trial program. So yeah, I would like to end this section sharing that for all ABCAM products, you can have a peace of mind using them because they are covered under our product promise guarantee for 12 months from the date of receipt. We guarantee that this product will work in the application or species listed on the data sheet. And if it's not, we are happy to provide technical support to help you troubleshoot the issue, or also to provide a suitable resolution such as a free replacement in the event that the antibody is really not working. And as previously mentioned, if if you are unable to find a product that is validated for your target application or species, we also have our ABCAM trial product to help you. So you just need to let us know, or let Apical know, which product you would like to test in and whether is it an untested species or application, and we will help to check the eligibility. And once the eligibility is confirmed, you can proceed to purchase the product and do the testing. So what we need you to do is actually to share the product, share a review with us on how the product worked in your experiment. And once this review is published and submitted and published, we will then give you a discount code that is equivalent to the full price of the product that you tested. So in a way, it's a free antibody, but you have to first test it and submit a review. And moving on to the next section, I would like to briefly talk about the things to note when transitioning from manual to automated IHC staining. So this slide actually gives a good summary of the main advantages of performing IHC staining using automated platforms over the traditional manual IHC procedures. Due to the accurate and precise control of temperature and incubation conditions, automated IHC makes standardization easy while minimizing human error. So there is a greater consistency and reproducibility. It also provides for flexible simultaneous antigen retrieval protocol, which further allow for quicker optimization and high throughput IHC staining. So when transitioning from the manual IHC to automated IHC, the two things that you would need to consider would usually be the heat-induced epitope retrieval method, and as well as your primary antibody condition. So in manual IHC, the antigen retrieval method is usually using the microwave or pressure cooker. However, in the automated system, the slides are now heated on individual station. So although the heating methods are slightly different in manual and automated IHC, it can be easily translated since the machine usually allows you to choose the optimal time and temperature. And there's like a wide range of temperature that can be used. So this is usually easily translated. But for primary antibody incubation, if let's say the antibodies that you are using are done at room temperature for 1 to 3 hours, they will be easily adapted onto the automated platform. However, if you are using like overnight incubation of your primary antibody, this can be usually hard to translate it to the automated system due to instrumental limitation. So oftentimes it might not be possible to do overnight incubation at 4 degrees when you're using an automated machine. So to get the same low specific binding at 4 degrees but with a higher temperature, you might need to test a range of primary antibody concentration, or as well as add in the additional blocking step or modifying the antigen retrieval step. So we are happy to share with you the automated IHC protocol using various platforms such as the Leica Bond RX or the Ventana Ultra that you can use as a reference when you are doing the automated IHC staining. And just to give an example, using AB225260, which is a antibody targeting IBA1. So in manual IHC, the heat-induced antigen retrieval is actually done using a citrate buffer at pH 6 for 20 minutes, followed by the overnight incubation at 4 degrees. But when you translate it to the automated staining, you can see that actually we need to use a longer antigen retrieval time so that the staining intensity will still be the same when we change the primary antibody incubation time to 1 hour at room temperature. So therefore, you really need to play around with the antigen retrieval condition and the time, as well as the primary antibody concentration when changing from automated IHC to achieve the optimal staining. So yeah, therefore, yeah, I would still like to highlight that if possible, then you should select antibodies that are tested for multiplex IHC or bond RX validated, as they will have the condition, they have been tested for a shorter incubation period in our in-house testing, and all the data for the condition used will be included on the data sheet that you can use as a reference. And yeah, so now we have come to the last part of our discussion today, which is troubleshooting and optimization tips for IHC. And I will also end off this seminar by sharing some resources that are available on our website. To troubleshoot, the first and foremost is that it's very important to know some basic information about your samples and protein targets. So for example, what is the protein expression level in your samples, and where is the subcellular location of the protein? What treatments were these samples subjected to, or the samples required in order for you to see the protein expression, such as if you are testing for phosphorylated proteins. And also, we recommend to include the controls to ensure the specificity of the staining. And let's say if you are doing multiplex staining, it is always important to include a single stain control for each marker before proceeding to your actual experiments. So the three most common issues that we usually hear or researcher typically face is first that there is no or weak signal, and second, it would be high background staining, and last but not least, it would be incorrect or off target signal. So for example, you are observing the staining of the membrane when you are expecting a nuclear staining. So for this part, the troubleshooting tips would be more for the traditional manual IHC method, but they can also be applied to multiplex staining. So let's take a look at this issue one by one. So for weak and or no staining issue, they can be due to your sample issue. So firstly, you will need to consider whether is it due to the lack of target proteins in the tissue samples. So for this, you can check the literature if you have not already done so in the planning stage, or by running a positive control. And if your target sample is present in your sample but is expressed in low amount, you may need to include an amplification step, such as using HRP polymer system, or try coupling it to the brightest fluorophore available for fluorescence detection. So as a general thumb of rule, if you are using fluorescence detection, you should always use the brightest fluorophore for the least abundant protein, and vice versa. And let's say if you are working with paraffin tissue section, you will need to ensure that the section are completely deparaffinized so that it does not interfere with staining. And the fixation duration might also need to be optimized to prevent over fixation of the sample, which may mask the antigen. So yeah, so let's say if the protein is located in the nucleus and the antibody cannot penetrate the nucleus, you might not be observing any staining as well. So we always suggest to include a permeabilization step using Triton X, even if you are using paraffin tissue sections, to allow for better penetration of your antibodies. And also, Yeah, so if the protein is a membrane protein and permeabilization has damaged the cell membrane, it might also lead to a lack of signal. So for this, you can consider using a less stringent detergent such as Twin 20 instead of Triton X. So Triton X is more for when you are intending to detect nucleus protein. And yeah, so other times no staining can also be due to reagent issue. So it's always important to ensure that the primary and secondary antibodies are compatible with each other. And if there is too little antibodies, if the antibody concentration is too low, then you can consider increasing the antibody concentration or incubating it for a longer period of time. You may also want to check the antibody product data sheet to make sure that it is suitable for the specific type of samples that you are working with. So let's say some antibodies may not detect the target protein in the native 3D conformation. So if you are using IHC, then yeah, this antibody might not be suitable. So always ensure that the antibody that you are using is validated for the application of interest. And yep, so also you will want to make sure that you store the antibodies properly so they do not lose activity due to improper storage. For example, if you are performing fluorescence detection, you need to ensure that the fluorophore conjugated antibodies are stored correctly as recommended. So please always refer to the datasheet for the storage condition of your antibodies. On the other hand, for high background staining, the first thing we need to check if this could be due to insufficient blocking and whether you will need to use a longer blocking time. And it might also be necessary to use a different blocking buffer. We usually recommend using normal serum of the species of the secondary antibody as it gives a stronger blocking as compared to using BSA. So, yep. So, yeah. And contrary to no staining issue, you can also check to see if you have used too high a concentration of your antibody, as that could also lead to high background. And let's say if you're using TSA or OPA reagent, usually they are much more sensitive as compared to the traditional fluorescence detection. So you might need to use a lower primary antibody concentration to avoid this high background issue. And high background could also be due to the non-specificity of your secondary antibody. So we can always check to ensure that the secondary antibody is against a different species to your samples. And we also can run a no primary control to check the specificity of the secondary antibody. Alternatively, we can also use pre-adsorbed secondary antibodies that can help to prevent non-specific binding issue. And sometimes if the tissue are not washed sufficiently, there can be residual fixative or unbound antibodies that remains that produces a background signal. So in this case, you could increase the number of times of washing or the duration, as well as to add detergents such as 0.1 Triton X or Twin 20 into the washing buffer so that all the unbound antibodies can be washed off. And last but not least, the third common issue is that we encounter incorrect signal or non-specific staining. So if you find an uneven staining pattern, such as in this example here, this could be due to insufficient deparaffinization. So we need to ensure that fresh xylene and longer time for the deparaffinization step is being used. And we also need to check if we have blocked the endogenous peroxidase, especially if we are using chromogenic detection. But let's say if you are using TSA detection method, then this would not be as essential. And also, if there is high unspecific staining on the edges, such as in this sample here, then this could be due to the tissue sections being dried out during the staining process. So to avoid this, a humidified chamber can be used. And last but not least, sometimes incorrect staining can also be due to too high a concentration of primary antibodies being used. So to avoid these, we always recommend to try a different dilution of the antibodies in your initial staining to ensure that you determine the optimal concentration to use. So yeah, I would like to briefly end off this section with some kits that we have available. So we do also have multicolor enzymatic IHC available. So for all these kits here is for double staining. So and we also have the kits available for triple staining. So yeah, so if you are looking at a multicolor enzymatic IHC, you may want to consider getting this kit as they have been optimized for the multi-marker detection. And also, if you are working on thicker section, clearing kit will also be a useful tool, as this can help to prevent as like thick sections usually are affected and limited by light scattering. So you can use this tissue clearing kit to help to increase the tissue transparency so that you can get a clearer staining. And last but not least, I'll just like to share that we also have many protocols and resource page available for various experiments and application on our website. So you can just type the application you are interested in and then see what resources are available. And of course, if you encounter any issues that requires further assistance, you can always approach our authorized distributor, Apical, and they will be happy to assist you. Or you can also reach out to us directly via a chat or an email, and our team at ABCAM, will be happy to assist you in terms of troubleshooting, or if you need help in product search or advice, we can also help you with that. So with that, I have come to the end of this seminar, and I'm happy to take on any questions that you have. Thank you so much.